Yeah. All right, so this is midterm review day 4. It'll be posted up online. If you have any particular questions, don't email me, because I probably won't see them in time for you to get everything done. I'd like drop by after school on Monday or Tuesday or something like that and ask me questions if you got those. You will also have a literally a study. You will actually have a study session with me before you take your exam. It's about 50 minutes long. So if you have particular questions, I'd love to help you out with that. But I'm not running a whole review session that day. It's like a spot check. Hey, help me with this. Help me do that. I'm going to study. No, you're not going to like fiddle-faddle, whatever that is. You're not going to fiddle-faddle. What?
There's popcorn called fiddle-faddle.
Yeah, you won't be popcorning either.
I've never heard that in my life.
Explicitly no popcorning here. Okay, so where did we leave off yesterday? Can somebody remind me?
Binary fission.
Really? Yeah.
Okay. Cool.
Yeah, diploid, haploid, binary.
Yeah. Okay, so I'm now looking at your study guide. So we're just going to run through this thing super quick. And if you got particular questions, you know, that'd be nice. I'm going to try to hit everything that I possibly can. So we'll just go by the numbers here, okay? Number 1, 4 functions of cell division. It's going to be make diploids. Oh, this is going to take way too long. What?
Is this still chapter 12? Or what?
I believe chapter... Yes, I believe it is chapter 12. So it's going to make diploids. You are going to divide the cell. It's also going to preserve genetic info. I'm hearing a lot of fiddle-faddle. And I would also say that it prevents mistakes from replicating. Say it one more time.
What are you talking about?
Number 1 of your study guide.
I don't have it with me.
It's posted online for you, so you can get to it. If you watch the notes or anything, or you just want to match your numbers up as best you can, we can do that. Which cells divide by mitosis? Everybody should know this, right?
Yeah.
Body cells, sometimes called somatic cells. Notice how we're being more detailed on this this review here. Distinguish between the following terms and name the phase of the cell cycle in which you would find these structures: chromosomes, chromatids, centromere, and complementary strands. Oh boy. Oh boy. Oh, it's Chris. Could you be so nice to get that? Okay, so A says chromosomes. You're going to find chromosomes pretty much everywhere. Almost all stages will have that information. But B, chromatids separate. Chromatids separate. You remember how yesterday we were talking about how a chromosome looks like this? Yeah? And you see how 1/2 of this thing, that's called a chromatid? Well, that chromatid will separate during anaphase, right? I mean, I guess as a result, you'll see telophase having it too, but chromatids would be showing up in anaphase. Centromere, this is going to be during prophase and metaphase. Okay, is my handwriting too sloppy? We okay? You following along? Okay, complementary strands of DNA. In the cell cycle, I would tell you that you want to look at something called synthesis. That's where you'd find that stuff, because it's going to be a copied strand of DNA, complementary strands. You doing okay? Any questions there? No? Okay. Go ahead.
Number. Letter C, subprophase and metaphase.
Yes, ma'am. All right, what happens during G1, S, G2, and gap 0 of interphase? Do you guys know what gap really kind of means?
Growth.
Yeah. You should know gap equals growth. So, hey, you now know what happens during gap 1 and gap 2. Gap 0, they're not really going to grow. I'm just going to go ahead and distinguish for you. Gap 0 means it's just, it's what's the word? Senescence. So it doesn't change. Oh boy. And then we have synthesis. You should know synthesis means we duplicate DNA. You doing okay? Okay, 5. Name the major events of prophase, metaphase, anaphase, telophase. Did we do that yesterday? I think we did. Did I draw out the stages?
Yeah.
Okay, so I'm going to skip 5 because we kind of did it yesterday. 6. How is cytokinesis different in animal cells and plant cells? I think we talked about this. There's something called the cleavage furrow. I can't spell. That's going to equal animals, and then we have the cell plate. Yes, that forms in plants. I'm bad at spelling. Yay! Doing okay? What? Who says I don't teach in English? Number 7. How are the following involved in cell division? Oh boy. Oh boy. Shh. We're running out of time. Number 7. Surface to volume ratio. Remember how the size of a cell really matters, right? There's actually a whole lab that you could kind of do on this whole thing. But instead of having one gigantic cell, we would prefer to have one gigantic cell split into much smaller cells. So we want to have a particular ratio. Can I remember that number right now? No. I think it's 4:1, but I'm probably 1 there. But you should look that up. We prefer this. That's why we don't go with just gigantic cells. There's a certain size limit that cells can have. You got to keep that in mind. I'm sorry I can't remember the number. I'm rushing. Density-dependent inhibition. So the number of organisms that are like cells that are stuck together, that determines what they can do. Remember how sometimes you have to reach a threshold? Remember the biofilms that are moving around? Like the plaque on your teeth, they crawl around? Or some of those fungi that basically, or molds, if they grow on a tree and it's too dry, they can like form like a little bubble and kind of roll off and fly away. You guys remember that? Quorum sensing, do you remember that? Kind of. Quorum Sensing, we even watched an entire TED Talk on it. Yeah, that whole thing. So that's what you should keep in mind there, Quorum Sensing.
What is that picture of?
This, of like one cell, and then like how the same volume of cells, we would add surface area to it by dividing it so many times. So we prefer smaller cells, more cells of more surface area, instead of just one large cell with the same amount of volume. So we prefer having a higher ratio. Hey, should be a higher ratio of surface area to volume. Go ahead. You sure?
It's Quorum Sensing?
Yes.
Okay, just make
Sorry, handwriting is awful. It will be awful today. C, checkpoints. Well, hey, we got to check for mistakes, right? We said there's 3 areas. I think yesterday I drew the cell cycle and I said there's these 3 boxes. Those areas are where they occur. So you should be aware of those. Can I move on to meiosis? Yeah? Okay. I'm just going to label this meiosis. Oh boy, I'm rushing. Which cells divide by meiosis? What are they? What is this, number 8? What was that? Sex cells. They also have another name. Gametes. Gametes. Okay. 9, meiosis 1. List the phases of meiosis 1 and include the following terms as you describe the phases. Oh my gosh. Which ones are you having a hard time with? A through H. Which ones do you want me to do? Pick a couple.
N would be cool.
N?
M, like.
For number 9?
Wait, number, sorry. I got 2 ahead. Sorry.
Can you do A?
A, reduction division. Reduction division, this means when you divide, or division causes a reduction in the number of chromosomes. Yeah, mitosis is not reduction division because it makes more of the exact same copies, right? Diploid makes 2 diploid cells. In meiosis, we go from 1 diploid cell to 4 haploid cells. So we're reducing the amount of DNA that's found in each of those. That's why we consider it reduction division. And technically, that'd be meiosis 2. Go ahead. F. F would be tetrads. Okay, so let's do, I'll do F and then D. Tetrads are when homologous chromosomes, we'll say like hug each other. So when we draw that out, this is really important for the process of crossing over. So we have a homologous chromosome there. We have a homologous chromosome here, and pretend they're like actually hugging each other, like really tight. That's where we can have those chiasmata. Do you guys remember those? Like the exchange of genetic information there. It's like getting tied up and they pull apart and they have a little bit of each other that's crossing over. That's what a tetrad is. So the homologous chromosomes laying on top of each other, forming like a super chromosome. D says 2N. 2N means chromosome number, so that's the diploid chromosome number. So we have the number of chromosomes of 23, so that tells us we have 46 chromosomes. Okay, that's the number of chromosomes humans have. Anything else? Are we good? You sure? G, synapses. Synapses is going to be that connection. Oh boy. Yeah, I'm drawing it. Deal with it. Let's pretend those are, whoops. Let's pretend those are the connections of those homologous chromosomes. That merging that forms right here when when that genetic info merges with the other homologous chromosome. That would be synapses. Can I move on? Okay, let's hit up 10. Meiosis 2, list the phases of mitosis 2 and include the following terms as you describe this. Okay, cool. Let's do this. I, P, M, A, T, C, P, M, A, T, C. We got prophase, metaphase, anaphase, telophase 1, prophase, metaphase, anaphase 2. This is all going to be including meiosis 2, and this will be meiosis 1. What we're saying in meiosis 2 here is that haploids form. Well, haploids will form after this point. Everything is now going to be a haploid. What's up?
Can a kid come up and grab his wallet in case he left it in here?
Anybody see a wallet?
Unless it's in that jacket.
No, that was left 2 days ago. Sure, but they won't find it. But go ahead. 1N, that's actually how we represent what a haploid is. That is 1N. Honestly, we usually just write N to describe the chromosome number because it's just 23 in this case, half the amount of DNA that would be found in a diploid cell. And then we have sister chromatids. Sister chromatids are the 2 halves of a chromosome. So if we were to split this, that's a sister and that's a sister chromatid. Okay, so the 2 halves. You with me? Yes, cis-tid. Feels like I'm saying something dirty, but I'm not. Number 11, which process produces genetic variation, recombination, mitosis, or meiosis? That should be an easy one. It's meiosis. Genetic recombination, crossing over, and independent assortment, they all enhance genetic variation. That's the purpose behind doing all that. You with me? Yas? You good? I'm seriously like having a small heart attack because I drank coffee this morning and I'm super nervous about our timing.
It's okay to You want to throw a Red Bull in there too? That might make it better.
No, I'll do that. I'll do that later. Okay, so we have all hereditary to speak of. Can I move on? You what? Is this a Yeti? It's a cup? Yeah? Why?
Yeti brand?
Yeah.
Okay.
Yeah, it is. Thanks. So, hereditary, number 12.
Hey.
Hey.
Now can you talk about
You know what? I probably will. Yeah. Does anybody have any letters before M you want me to go over? A locus. If you look at a gene, like let's say that's a chromosome, the placement of a trait, that is called the locus. So the placement or position. Okay. Anybody else need a thing?
M.
Everybody wants M, but anything before M, because there's quite a bit there. G and H, phenotype and genotype. Good question. Which one is that? G and H? Okay. G says not appropriately. It should have been genotype. So phenotype, pheno equals physical appearance. Wow, I can't spell. I don't know who that is. Oh yeah, it's this kid.
Why did you put his face in?
I don't think it's in here, but you can take a peek if you want to.
I think so.
Natalie, Justine, this kid's probably going to look for a wallet near you. So it's the physical APPEARANC. It's a physical appearance of the organism. So, Caitlin, you might actually remember this. No problem. If you have big A, that tells you if it is going to be homozygous dominant, right? What's it going to appear as? If we say big A is going to be like, I don't know, let's say like clown feet. Creepy. And little a is normal feet.
Did you just say clever feet?
Clown feet. So this person here would have clown feet.
You guys were big.
So this would be the phenotype.
Die.
Only trying to help you. Only trying to help you. And then the next one, H, is going to be genotype. Genotype is the, we'll just say which alleles the organism has. That is the genotype, the two-letter combo. So genotype, phenotype. Genotype, the codes, the genes, the two alleles, and then the phenotype are the physical appearances of those genes or alleles. Doing okay?
K and L.
Okay, M. K?
K and L. Can we do
Oh, K and L, sorry.
As long as you get to
Monohybrid cross. Monohybrid. Bless you. That's a monohybrid cross when you just use a normal Punnett square. It's one letter combo that's crossed up. It doesn't matter what the genotypes are. It's just a simple Punnett square. It's called a monohybrid cross because it's one allele set at a time, one genotype. L is going to be a dihybrid cross, so that means it's two alleles that are inherited together. So this would be like either that super Punnett square. So it's two traits at the same time. So we usually would refer to a parent that's written like this as something like this, two letters at the same time, and you'd cross that with a parent with still the same alleles. Of course, they might have different genotypes, but they'd have the same alleles available to them. Yeah.
Is allele just the single, like one letter?
Yeah, an allele is just a single letter. So you have to have two copies of alleles.
Okay.
So that's why, yeah. Okay, finally the most desired one here. M. I'm going to skip it. Let's do a different letter.
stop it.
We've got P, F1, and P, F1, and F2 generations. Let's go back to Mendel's experiment. It does a great job on showing this. The P generation equals when he takes a purebred, like let's say it was a white flower and a purple flower. Let's go and change the letters. Let's say big P, that's purple, and then little p. Those get crossed together, right? So they cross together, and we get a result. All those kids are going to be big P, little p. Do you guys know that? Okay. This generation, if we cross it with another kid of that same generation, that's called the first filial generation, which is a cross of the children with each other. I know that sounds super gross and nasty, but it's not with plants. It's fine. And then F2 generation is when you cross these again. So whatever the byproduct is, or actually whatever is created from this would be that. So you'd see some would be born this way, some would be born this way, and some would be born this way. The cool thing is we realized it creates a 1 to 2 to 1 ratio, and also that's a genotypic ratio, but when it comes to a phenotypic ratio, that makes a 3 to 1 ratio. So most of the kids show the dominant trait. You with me? So I did M. Do you guys have any questions? Sure. I'll squeeze it in the middle here. N, test cross. Let's say you've got some unknown organism. You just know its phenotype. Let's say it's purple. You know that could be big A, or big A, little a. So how do you figure out which one it is? You do a test cross, which means you do a Punnett square, and you cross it, or actually you wouldn't just do a Punnett square, you'd actually mate this thing with the other organism, and you'd mate it with always a homozygous recessive organism. Sorry. Oh, no, we did it right. You'd always mate it with this. So if it's big A, all your kids will look dominant. So that tells you that you have a homozygous dominant parent. If it's big A, little a, you'll end up with all of, not all of your kids, but half your kids will look homozygous recessive. So it kind of is the test to find out if you have a heterozygote or a homozygous. Scanning the room for questions. My heart rate is definitely up. All right, I'm moving on. Not enough. Now you're all hoping for a heart attack. But that's not true, because I would have taught you everything, and the copy of the midterm's already there.
It's a traumatic experience. I can't.
But the test is still there. My dream would be for you to take it and pass it.
Well,
You're gonna honor my dreams, right, Rachel?
no.
Shut up, Ashley. Okay.
Yes, we can Bernie's you if you die.
That's probably true. Okay. 13. Describe the following rules and laws of Mendel's and give examples of each. Let's ignore A. No, because it won't show up on your test. Unless you want it to show up on your test. And be nice, Caitlin. Dominance, the law of dominance. That means big A beats little a. That's the law of dominance. There's a dominant allele and a recessive allele. If they're ever together, big letter wins. Does that make sense? Okay. Is that always true? No. But, hey, it's a thing that he didn't think of, but somebody thought of, and credit it to him. Sorry. C, segregation. When we have two letters, either big A, little a, or little a, no matter what, they will always separate when gametes form. Oh, my gosh. Only one allele is sent. Okay? So when gametes form, we only send one of those letters, and it's random, lucky for us, which actually leads us to the next thing, D, independent assortment. Alleles, and in turn chromosomes. Formation doesn't depend on others. So if we look at that, okay, let's try it this way. I've drawn this before. All your whispers are making me extra nervous.
Olsen, chill out, bud. Because we're talking about you.
Probably. No, you guys keep asking, like, what time is it? You're making me nervous.
We have 10 minutes.
So, if we look at this thing, Independent assortment, it could go one of two ways. For just these simple chromosome combinations, we're saying the big chromosome, because there's a red and a blue for each, the big chromosome doesn't affect where the little chromosome goes, does it? If you look in example 1 over here, it shows that we split the cell this way, and all red moved to the left and all blue moved to the right. In example number 2 over here, we can see that we split it so that a big red and a little blue go on the left side, and a big blue and a little red go on the right side. This is independent assortment. Whichever chromosome it is doesn't affect what the other chromosomes do. Does that make sense?
Sense. Yes.
Thank you for saying sense. Can I move on?
Yeah.
Okay. Number 14. We're in blue now. Cool. Do you guys need me to define or show you any examples of some of these things, or? Okay. I can help you really easy with that one too. Let me define some of the ones that I feel like you probably don't know. Does anybody know what epistasis is? Good. Epistasis. It's the suppression. I will only put question. I will tell you when something won't show up. How about that? It's possible this could show up. You with me? If I'm defining it, yeah, it's a possibility. So suppression of one gene by another gene. Suppression. Did I spell it right?
Yeah, you spelled it right, but like, what do you mean by that?
We're saying like, hey, if you have, okay, let's say it's like hair color, blonde hair. Oh my gosh, blonde hair, brown hair, red hair, whatever hair. There could be a gene that says, no, you're now going to be gray hair. Oh, so everybody with gray hair seems to have like gray hair. It's not like a weird shade of gray. Everybody seems to have gray or white hair, right? So we're saying that expression of that gene no longer being suppressed because another gene saying, nuh-uh, I take over now.
Oh, okay.
You guys might have a hard one with E, I'm guessing.
Yes.
Yeah? It's funny that nobody's asking me these questions. Good. One gene basically can affect affect?
Yeah.
Look at me. Can affect more than one phenotype. Good job. Or phenotypic traits, but one phenotype. Another one would probably be the next one, F. Polygenic inheritance. When one trait is controlled by two or more genes. So we're saying one trait, you would need multiple different alleles to determine what that is. A good example would be something like skin color, okay? Because there's not just like, we might say a person is black, they're brown, they're white. No, that's not really what skin color is. It's like a rainbow of colors. And basically, if you have an, I think, okay, I'm going from memory here, but I'm pretty sure your book does a good example of this. And I think it shows a trihybrid cross, which is like a huge box. And it shows you 3 different alleles involved, A, B, and C. That would be 6 different alleles involved at the same time. And if you have dominance on some of them, that just adds to the number or the amount of melanin that's going to be produced in the skin. So the more dominance you have, the darker the skin would be. The fewer dominant traits you'd have, the less melanin you'd have in the skin, so it'd be a lighter tone. But yeah, that'd be a really good example of that. I will tell you, this one, don't worry about it. It's not going to show up on your test. Congratulations. X in activation. No, J would show up. Non-disjunction. Homologous chromosomes don't separate. This would be like a chromosomal abnormality. If you guys remember us talking about omies, like trisomony, like trisomony 21, Down syndrome. We're saying that chromosome isn't separating it, so an extra copy ends up in that cell, which means that individual would have an extra copy of that chromosome, causing that condition. Any others? Are there any particular vocabulary words you want me to define for you?
I already made breakfast.
Okay, cool.
I made flashcards.
Congrats.
Yeah.
Why did you tell me congrats?
Because you were critiquing me earlier.
I was not.
I'm pretty sure you were.
When did I critique you?
Earlier.
Earlier.
Yeah, you said you were doing that.
I did not say that.
That's what I mean.
Why do you want me to have a heart attack?
I literally was not mean.
I've been recording this entire conversation.